Actually, I didn't really know what to talk about, especially after the first two talks, which were so elegantly delivered. One on work on wildlife, and the second is work with the society on Clip. So I've got nothing, that exciting to talk about. So I thought maybe some people are asking, what is it that she has done in 29 years in UKM? So basically, what I'm going to tell you today is a history of my journey through UKM and how I have reinvented myself from what I trained in originally to what I am doing right now. Okay, so let me start with a little bit of history. As most of you who are in this session today, I am also an alumni of UKM, okay, from the biochemistry program. I graduated in August of 1998. So when Provani said veteran just now, she wasn't kidding, right? That's old. Right, so partway through the final year of my time in UKM, I decided that it would be interesting to learn more about molecular biology, which was only then coming up in Malaysia. So I I applied to enroll in a one-year intensive MSc program at the University of Sussex on molecular biology and genetic manipulation. So I managed to get a scholarship from the British government. This is the Chevening program. And also a second scholarship from Kwok Foundation, which allowed me to spend a year in the UK. So partway through my research project, my supervisor asked me if I would like to stay on to do a DPhil. So in Sussex, PhD is referred to as the DPhil. And so I said yes, and of course, and I had to look for funds again. So I was lucky enough to be given a Chevening scholarship for the second time. I don't think anybody else has achieved that. And I also managed to secure a scholarship, which was awarded by the Council of Vice Chancellors and Principals of UK Universities, CVCP, which is now known as Universities UK, and a small stipend from my supervisor's Wellcome Trust grant. But that funding was only enough for 2 years of my PhD. So, I had to find money for at least the third year. Luckily enough, when I wrote back to UKM, UKM was very willing to offer me a scholarship to cover my third year. And I promised to come back and work for the rest of my life at UKM. So that's how I came back in February of 1993. So I'm sure most of us have the same idea when you come back and you report for duty. All right, so you have to think about your research project and how you're going to establish yourself. And usually the first thought that comes to mind is, okay, let's continue on what you were trained on. So that was the first intention, which was, okay, maybe I didn't mention it. So for my PhD project, I worked on transcription-coupled DNA repair in human disorders. So totally different from what I'm working on now. So anyway, at that time, when I was thinking about continuing along that track, Dr. Hasida introduced me to her former classmate, Khalifah Siddiq at University Malaya, who had also trained in DNA repair. So together, we managed to obtain some funding from Torre, as well as through the IRPA mechanism. And we were interested in looking at the effect of UV disorders on the P53 regulatory protein. So we held consultations with Dr. Suraya Hani Hussein at HKL, who very kindly agreed to come on board. And then she provided us basically with, she's a dermatologist, so she provided us with skin biopsies from patients who were undergoing UVA treatment together with the drug Sorolin for skin disorders. So my PhD student, that was my first PhD student, Sok Cheng. So her job was to take these skin biopsies and to generate cultures. So basically fibroblast cultures, which she managed to do quite elegantly. And by spending over a year at St. George's Hospital in London in Dot Bennett's lab, she was able to maximize experience and expertise in generating and characterizing these cell lines. So basically the project involved this new technology at that time, which was single-stranded conformation polymorphism. So you extract DNA from the control cells as well as the cells that you're studying or the phenotype that you're studying. And then you run them on polyacrylamide gels, not agarose gels. These are vertical polyacrylamide gels. And we look for changes in the profile between the disease state and the wild type state. So that was quite an interesting project and it was successfully completed. But at the same time, when I returned to UKM, the head of department at that time, the late Professor Osman Omar, he told me not to work in isolation and it would be best to join an existing research group. So what did I do? The easiest solution to that was to rejoin the melioidosis group, because I actually did my final year project on melioidosis, specifically when it was still called the bacteria was still called Pseudomonas pseudomallei on the exotoxin under the supervision of Dr. Rama Mohammed. Okay, so just a little bit of background information. What is Burkholderia pseudomallei and what is melioidosis? So Burkholderia pseudomallei is the causative agent of melioidosis. It's a bacteria, it's a gram-negative bacteria, which is found predominantly in all countries that span the equator, because it thrives in warm, moist soils as well as water bodies. And it can easily enter the lungs or through open wounds. That's why those particularly at risk are, for example, farmers, paddy workers, and so on. Even, anybody playing football without wearing boots or shoes and has a cut, there is a high probability that you could get infected. Okay, this disease is basically, even in Malaysia, it's non-notifiable. So there are no exact records of what the incidence rate is. But a calculation that was done on a sort of a global basis hinted that there might be up to 165,000 cases per year, of which more than 58% are going to die. So when you look at the fatality rate, you can see that it is significantly much higher than even dengue or malaria. Right, so this is a very unique pathogen and a very unique disease to be studied. All right, so when I joined the group, okay, I was given a project to continue on Prof. Rama's sabbatical project, where she had generated hybridoma cells with the expectation that these hybridomas are going to produce monoclonal antibodies against the exotoxin that I had been studying during my final year project. So for those of you who are not familiar with hybridomas, essentially what you do is you challenge mice with the antigen, then you obtain the spleen, you produce the spleen cells and fuse them to myeloma cells. So these are cancer cells which will provide more, immortality to the spleen cells so they can live forever. As a result of the fusion, you get your hybridomas, but these are a mixed population of cells which produce all sorts of different monoclonal antibodies. So when you are looking for antibodies that specifically target one particular protein of interest, so in this case, it was the exotoxin, the next step is called dilution cloning. So with dilution cloning, the idea here is that you have to get individual cells, and you then have to screen these individual hybridoma cells against your target protein, so that you then end up with monoclonal antibodies which are specific to your protein of interest. Okay, so that was my job. So my students went on to carry the work that I had started, and we successfully produced these monoclonals against the exotoxin and characterized them. Now, at about that time, okay, in sort of late 2015, late 1995, early 1996, there was a new technology that we had read about. And this technology involved bypassing the need for producing hybridoma cells to make monoclonal antibodies, but instead you could make recombinant monoclonal antibodies. And so we thought that this was a good way to move forward from the traditional into the more current methodology. So essentially, it's called antibody engineering using phage display. So again, you do have to start off by immunizing mice with your antigen of interest, and then you harvest either the liver or the spleen, you extract RNA, you convert it to cDNA, and then you amplify a repertoire of antibody genes. So you have a whole set of primers that are meant to cover as many different combinations of your antibody genes, and you put them into a single PCR reaction so that you get multiple different single chain or FAB genes being amplified. So the amplified genes are then cloned into a phage-mid vector, which is then transformed into E. coli. So what happens next is that you now have a library of these phage-mid which are carrying different antibodies. So again, this is mimicking the situation of the hybridoma cells just now, where you have multiple different antibodies. So the next step is something which is called panning, which essentially you're trying to enrich for just the clones that carry the antibody of interest towards your antigen. So this is a step that goes through multiple cycles, usually up to 4 cycles, so that every step enriches for the population of antibodies that you're interested in. So it's a fairly complicated sort of protocol or approach to start without any prior knowledge. So what I did was I went to Cold Spring Harbor in upstate New York to participate in the antibody engineering workshop, which is conducted by these 3 guys from San Diego. Okay, so that was a 2-week intensive workshop. And when I got back, I thought that this was, yes, an interesting technology that we could take further. And so I asked Carlos if he would accept me for a sabbatical in his lab, and he was very happy to do so. So armed with, I managed to obtain a Fulbright scholarship. And with that, I arrived at the Barber's lab in October of 1997 and was assigned to work with Christoph, one of the senior postdocs. So basically, the project that I had planned was, again, a protein secreted by Burkholderia pseudomallei, but this time it's a protease, because our initial work had shown that the protease digests the antibodies that the infected host secretes to try and fight off the infection. So the fact that this pathogen can actually, damage and destroy these antibodies is a unique way of how the pathogen manages to survive. And that's why the infected individuals can die as fast as 48 hours after infection. So basically, my project would be to make these recombinant antibodies against the particular protease. Okay, but when I arrived, okay, Carlos asked me to be involved in 2 other projects that were being run at the time. So one of it was Christoph's own project, which was, he was trying to make, his ultimate goal was to make humanized recombinant antibodies against a particular antigen of colon cancer. Okay, so the antibodies that we make using the phage display mechanism that was developed at the Scripps is we make chimeric antibodies. So what you see in yellow is derived from mice. Okay, what you see in red is actually the human backbone of immunoglobulins. So if you were to go along the route of the hybridoma technology where the antibodies are directly obtained from the mouse, and then you try to use this for therapeutic purposes, you're going to get into trouble because humans do not like antibodies which were not generated by themselves, so they would then go on to destroy the mouse antibodies. So one way of overcoming that is to produce chimeric antibodies. So where you just take either the single chain or the FAB component, which are the important parts of this whole antibody molecule, and you fuse them to a backbone of human. So now it's about 80% human and 20% mouse. So that's more acceptable to be used for therapeutic purposes. So what Christoph was trying to do was to convert these chimeric antibodies to make them even more mimicking human recombinant antibodies. So that's what's referred to as humanized. So my job was to create the library of the chimeric antibodies. And once I'd done that, then he took over and he went on to produce the humanized antibodies. The third project was being performed at the Burnham Institute, which was across the road from Scripps. And the people involved were Renata Pasqualini and Wadi Arab. So I actually met Renata in Cold Spring Harbor. She was one of the other participants. And their idea was, so this sort of screening or panning is done in vitro in 96 well plate. But her idea was, can we do it in vivo? Okay, so specifically, can we find antibodies that home into cancer antigens, which her interest is in cancer. So again, I constructed the antibodies libraries for them. And what they did was, what Renata did then was to inject it into the mice. Okay, these mice had been genetically modified to produce tumors. So in this case, it was Kaposi sarcoma. And the idea was, could we find or could we isolate antibodies which would bind directly to the antigens of Kaposi sarcoma? So inject the library, wait for them to bind, then the tumor was harvested, was homogenized. And then I took it back to Scripps, isolated the enriched phage and then the next day we would proceed with the same procedure again. And again, this would go on for 3 or 4 rounds of panning. Okay, so it was quite interesting over 7 months working on all these different projects. However, it was not all work and no play. So I managed to do quite a lot of trips on my own or with the lab. Okay, one of the most exciting one was every year the Babas lab in February goes on a whale watching trip. So this is essentially off the coast of Mexico to watch whales and dolphins and so on. Okay, so once I finished my sabbatical and returned to UKM, how did I transfer that expertise? So initially, I had already been working with Prof. Zali at So we had been using a simpler system of phage display to look at binders towards the envelope protein of dengue virus. So in addition to that, I worked with Prof. Wan, okay, where we generated recombinant antibodies against surface proteins of the human parasite Toxoplasma gondii. And in a later project, we did peptide display. So with peptide display, basically you're replacing the antibody, the repertoire of antibody genes with a repertoire of random peptides. They are 12 mers. So you get many different peptides being displayed by many different phages, and you can screen for binders. So this technology was also utilized in collaboration with Tan Weng Xiang and Khatijah Yusof at UPM with Newcastle disease virus and also hepatitis virus. And the final project collaboration was with LGM, or Lembaga Getah Malaysia, where I worked with Dr. Sundaresan. And he was interested to see if we could produce antibodies that would inhibit the toxin that was produced by a fungus that was basically destroying the rubber leaves. So it was quite fulfilling when we managed to produce the library of antibodies, which were then subcloned into a vector that was more compatible with transforming into the rubber plants. And we actually had very nice evidence of the rubber tree leaves being protected by the antibodies that had been produced. So well and good. Then the next reinvention came about in 2005, when MOSTI decided that they were interested in expanding the existing brain gain program. So for those of you who are not familiar, the brain gain program was initiated initially to try and lure back Malaysian scientists who were working abroad so that they would come back, bring their technology with them, and establish their research group in Malaysian universities. So that didn't quite pick up or take off. So the next step was, could we get a collaboration between a Malaysian scientist in a foreign lab with local scientists in a Malaysian lab? So one of those scientists identified was Tan Manwa, who was based at Stanford University. And so because his expertise was in host pathogen interaction of Pseudomonas aeruginosa, using the C. elegans model as the host. Okay, so I was invited by MOSTI to propose a project that could integrate his expertise, okay, and establish the use of C. elegans as an alternate model to mice or to cultured cells for studying host pathogen interaction. So ultimately, this interaction is analyzed through expression profiling. At that time, there was no RNA sequencing, so the platform for expression profiling was microarrays. So again, not knowing anything, of course, you can read, you can write about expression profiling, but if you don't know how to do it, it's very hard to lead a group. So I took myself off on a second sabbatical, this time funded with a fellowship from MOSTI, to Jim Adioka's lab at the Department of Pathology in Cambridge University. So he was working on host pathogen interaction of Toxoplasma gondii. And so I was able to conduct my own microarray experiments as well as the analysis. So the first time using R and bioconductor and generating volcano plots and box plots and so on. So now that I knew how these studies were conducted, so we submitted the proposal to understand why the infected host succumbs to infection. Okay, because as I mentioned earlier, melioidosis patients, particularly those at higher risk, can die as quickly as 1 to 2 days after infection, even before a diagnosis can be made. Okay, so we wanted to understand why the host succumbs so easily to infection, and to use both the C. elegans model as well as the mouse model as our infection host, because each one has its advantages, okay, and limitations. All right, so we decided to go with both models. So as Prof. Vani mentioned earlier, what we had to start off with was to set up a C. elegans research facility. So this is the first of its kind in Malaysia. And basically, having visited the lab at Stanford, essentially we duplicated everything that was in the lab at Stanford to do C. elegans work in my lab at the Macmal Biology Molecule. All right, in addition to that, we were also given all the worm resources that were available at Stanford, so which includes mutant strains, transgenic reporter strains, as well as 16,000 RNAi clones. Okay, so these RNAi clones were useful if you wanted to. So the worm has about 20,000 genes. So if at any point we wanted to make our own knockdowns or knockouts of any of the worm genes, then we had the 16,000 clones available. And so we were, one of MOSTI's mandates was that we were now the sort of referral center for anyone who was interested in working with C. elegans. So we provide worms, any strains that they want, any resources. They come to our lab to get trained on how to work with worms. All right, so what did we do? Basically, we had to start from scratch because it hadn't been done before. But anyway, so we start off with killing assays to find out whether Burkholderia pseudomallei can kill the worms and how quickly does it do it. So you can see from this chart that it kills very much faster than even Pseudomonas aeruginosa or something like Staph aureus, which is like if this is 1 day, this takes 2 days. It's quite a big difference. Okay, then I had a very good research assistant who managed to introduce the green fluorescent protein construct into Burkholderia pseudomallei, which was quite handy because the worm being transparent, so when you feed it with a bacteria or infect it with a bacteria that is fluorescing, you are able to follow the progression of the bacteria throughout the worm. Okay, so we got some very nice results. And in fact, we were featured on the cover of Virulence when our paper was published. Okay, so as I mentioned earlier, the main part of the project was to analyze the expression changes in expression of the infected host versus the uninfected host. So this is what we did with the C. elegans model. And I'm not going to go into any of the details, but basically just to highlight some of the key points. So when we analyze the microarray data, so we found that a large number of genes which were downregulated in the infected host were in fact genes that were under the control of promoters with the GATA motif. So one of the key transcription factors that binds to the GATA motif of promoter is the ALK2, and this has many homologs in humans, which are known as the GATA transcription factors. All right, when we did some fluorescence microscopy work to specifically interrogate the presence of the protein, the transcription factor protein, we found that in worms that were infected by Burkholderia pseudomallei, this protein had disappeared. There was no longer any ALK2 or GATA transcription factor. All right, and this was validated using Western blots. So based on that finding, we came up with this proposal. So if you look at the top part of the slide, this is a general picture that we observe, which is if any bacteria, a gram-positive or usually a gram-negative, when it infects the host, what happens is that one of the host response is to overexpress genes which are under the control of the promoter that has the GATA motif. And these include genes that encode for proteins which are responsible to lyse the bacteria, to detoxify any toxins that are produced by the bacteria. So basically it is overexpressing the immune response of the host in order to fight the bacteria. Okay, but the work that we did, the analysis that we did on Burkholderia pseudomallei, we were able to show that when BP infects the host, so in this case, C. elegans, what happens is that it uses its secretion system to secrete as of yet unknown effector molecule. What this effector molecule does is it tells the worm's ubiquitin protein system to destroy the GATA transcription. So I think most of you know that in human cells, once the proteins are expressed and then they complete their function and they are no longer required, then they are tagged and the proteolytic system will come and destroy those proteins because they're no longer required. But in this case, what happens is that BP specifically instructs the host UPS to destroy the transcription factor, and that's why we were not able to pick up any of the protein in the infected worm. And so in the absence of that particular transcription factor, all the genes which are downstream of promoters that carry the GATA motif, which was basically these genes earlier, are not expressed, and therefore there is a non-functional immune response. So you may ask, ALK2 is a worm transcription factor, but as I mentioned earlier, it has homologs in humans, which are the GATA transcription factors. So we expect that this is also happening in the infected humans. Okay, so we also did similar studies in mice, right? The limitation of the C. elegans system is that we can only interrogate the first line of defense, which is the primary immune response. So we can't really see the secondary immune response. And for that, we still have to depend on working with mice. So, but the nice thing was that the work on mice validated a lot of the information that we got with C. elegans, and in fact gave us additional information that showed how the bacteria is able to destabilize the inflammatory response. And also what it does is it shuts down this thing called MAC, which is part of the, what's that, complement pathway. So MAC is membrane attack complex, and the function of the MAC is to lyse the bacteria. So the fact that the MAC was being downregulated or inhibited by the bacteria meant that the bacteria could now escape any lysis, replicate, and then spread from cell to cell, causing a lot of tissue injury. And this was part of the reason why individuals succumbed very easily. So to follow up on this, another worrying part about melioidosis is that diabetics are highly susceptible to dying from melioidosis. In fact, I think you're aware that diabetes is a comorbidity that's associated with a lot of infectious diseases, tuberculosis, and even SARS or COVID. Okay, but the degree of how more dangerous or how more at risk you are if you have diabetes is way much higher for those infected by Burkholderia pseudomallei. So we extended this study in mice to mice that were made diabetic, okay, and then repeated the whole study again. And what was interesting was that, so these are essentially proteins which are involved in the immune system and the immune response. So what is shown in black is what we saw for the non-diabetic mice. Okay, you have expression as early as 16 hours post-infection and so on for most of these proteins. But what is represented by the gold bars here is the profile that we got from the diabetic mice. So you can see that in diabetic mice, some of these immune response proteins are not being produced at all. And for some of the others, there is a delay of up to 24 hours after infection. So this delayed activation provides ample time for the bacteria to replicate within the infected host and to spread quickly and to cause as much damage as it possibly can. Okay, so in the middle of that collaboration with Stanford University, we were also invited to propose a project to meet the call for proposals on a top-down basis. So members of PPBSPT decided to submit a joint project, and that was to look for potential biomarkers as indicators of diseases in fisheries and livestock. So we came up with a proposal that could incorporate members with different expertise and different scientific backgrounds. Okay, so we had 3 components to this project: coccidiosis, which is caused by Eimeria tenera, infection of chickens. And then we also looked at the parasite cryptocaryon irritants, which causes cryptocaryonosis in fish, such as sea bass. And we included Burkholderia pseudomallei as well. Okay, so it was a multi-pronged approach. Okay, this one had EST sequencing, this one had phage display and structural biology and so on. I'm just going to highlight the project that was done by Sylvia, which was on Burkholderia pseudomallei, which was looking at If you recall, what we had been studying with the Stanford collaboration was mainly looking at the host perspective. What happens to the host when it's infected by the bacteria? So in this instance, Sylvia was tasked with looking at what happens at the bacteria in terms of expression profile when it is actively infecting macrophage cells. So this again was basically developing new expertise and new protocols in the lab. In fact, when we first submitted the manuscript to BMC Genomics, they didn't believe that we could do it. And so it was rejected, okay? And only upon appeal did they finally agree to getting it published. So it was very nice work. And then she was able to show that at the early stages of infection, there were 20 odd genes that were overexpressed during the infection. Okay, and based on that information, she came up with this schematic where we were able to propose that when the bacteria, okay, which normally has flagella, when the bacteria is trying to enter the host cells, it camouflages itself by downregulating the production of lipopolysaccharide and flagella so that it is not recognized by the receptors on the host cell. Therefore, it can sort of sneak into the host cell, where at first it undergoes growth arrest. So it basically is staying silent because it has to acclimatize to the different types of stresses which are present in a human host. But once it has recovered, then it goes into full steam to produce various proteins and enzymes and toxins using the type 3 and the type 6 secretion systems. And this is what leads to the disease symptoms that we observe with melioidosis patients. So the question you may be asking is, so what has this got to do with biomarkers, since this project was funded as a biomarker discovery project? So anyway, the type 6 secretion system proteins were of interest to us. So Sylvia went on to produce the recombinant version of these proteins and screen them against melioidosis patient serum. And we showed that the one particular type 6 secretion system component was a very good antigen to screen for melioidosis. Okay. Right. So I'm now going to backtrack and come back to the Stanford project. So at the end of the initial 5-year period that we were given to do the project, so there was still a lot of funds available to us, and MOSTI was okay with the progress that we had conducted so far. So we were invited to propose another smaller project as part of the big project to utilize the available funds and to expand on potential research findings. So when we were thinking about what to do, I came back to this earlier small study that we had done with Prof. Rama. And this was a computational based analysis to identify Burkholderia pseudomonas essential genes. So essential meaning that if it didn't have any of the proteins encoded by its essential genes, then most likely the bacteria could not survive under different environments. So we also had to identify proteins which you do not have homologs in humans, okay? Because if eventually any of these essential proteins become drug targets, then you don't want the drug to be acting on the humans as well. So that initial project identified about 300 potential essential genes of the bacteria. So in the meanwhile, we had gone one step further, where we had done a genome-wide mutagenesis experiment using transposon-based mutagenesis for the purpose of identifying mutations which are involved in the bacteria's virulence or pathogenesis mechanism. So essentially, by introducing the transposons randomly, we were able to generate about 2,000 mutants. And they were picked individually and screened individually in C. elegans. Okay, so the idea being that we were going to focus on any of these mutants that rescued the worm from the infection. So rescue the worm means that, so this dotted line indicates the survival of worms that are infected by the wild type bacteria. And any survival curves which are shifted to the right of the wild type indicates that the worms are now surviving much longer because the potential virulence factor has been knocked out or interrupted by the insertion of the transposon. So we got from those 2,000 mutants, we were able to identify potentially about 90, which could be classified as virulence factors. Okay, so because we had this opportunity to propose another project, so we decided to take it on a larger scale. And for this, initiated a collaboration with Arnab Pine at KAUST in Saudi Arabia, okay, to use this technique, which is abbreviated as TRADIS. So TRADIS is transposon directed insertion site sequencing. And basically what it does is it incorporates the traditional transposon-based mutagenesis with high throughput sequencing. So you create a large library of transposon mutants, and now instead of picking them one by one and analyzing them, you sequence the whole pool together in the hope of identifying genes which are essential for the bacteria's survival. Okay, so the premise for this is that, so for example, if you have a transposon that's randomly inserted into gene A, okay, if gene A is non-essential, then you will have the bacteria that's carrying that deletion or insertion growing on your plate, no problem, okay? However, if it is essential, then that bacteria is not able to grow because due to the insertion of the transposon, the gene cannot be expressed, you don't get the protein, and therefore that protein is important for the survival, and therefore the bacteria does not survive. Okay, so the student on this project was able to generate a mutant pool of 500,000. Okay, so this was all pulled together and subjected to sequencing. And then the sequencing reads that were generated, they are mapped back to the bacteria's genome sequence, okay? And the analysis is based on wherever you get very low sequencing, number of sequencing reads, or you get gaps in the read pattern, okay? These are indicative of essential genes, okay? Why can't you get any reads here or here or here? Because the bacteria that were mutated in that particular gene, 2362 or 2363 and so on, they did not grow, okay? So this is a very different way of doing things. But as a result of that experiment, we were able to propose almost 500 genes which the bacteria requires at all times, okay, under any condition to survive. Most of these were involved in your typical core cellular functions, as you can see from this list here. And these genes were also evolutionarily conserved within other Burkholderia pseudomyli, as well as other Burkholderia species. Okay, right. So now that we have this library, what else can you do with it? So you can also use that same library to discover conditionally essential genes. So the term conditionally here refers to the fact that you can screen for which genes are now essential for the bacteria to survive under condition A or condition B or condition C, okay? And you might find that, or you'll most likely find that different proteins are required to are required for the bacteria to survive under different conditions. So we chose to go with identifying essential genes required for the bacteria's survival and adaptation in the infected host using C. elegans. So basically, the whole library of 500,000 mutants was fed to the worm, and then after certain time points, the worms are sacrificed. You isolate back all the bacteria, pull them together, extract DNA, and do the sequencing, and then you map the reads back to the BP genome. And not surprisingly, we found a lot of proteins which have already been identified. but almost 50% of these 144 genes were actually encoding for proteins of unknown function. So these proteins could essentially be novel virulence factors, or they could be serving other essential functions which make them ideal targets for the design of new drugs for therapy of melioidosis. Okay, so what else did we do with the C. elegans expertise that we now had? So again, another MOSTI top-down project. This time it was in a collaboration with IFAM, which is the Institute for Pharmaceuticals and Nutraceuticals based in Penang. So here the idea, again developed by Manua, was that we were going to look for anti-infectives. Okay, now anti-infectives broadly defined can be any molecule, whether it's a natural product or a synthetic molecule or a small molecule that inhibits the pathogen, or alternatively, it is one that does not target the pathogen, but in fact targets the host. Okay, so because we know that any new molecules which are introduced into the system that specifically target the bacteria don't have a very long shelf life, because the bacteria evolved to become resistant to them. So we were targeting more of molecules that can assist in the host immunity or host immune response. Okay, so we had two projects. IFAM took Pseudomonas aeruginosa as their model bacteria. So we worked on BP as well as on Staphylococcus aureus. Right, so the premise of this anti-infective screen is that if you were to add worms, pathogen, and your molecule of interest, okay, so this molecule of interest could be a natural product extract, it could be the active component, it could be a synthetic molecule, it could be a small molecule of some sort. So when you add all of them together, you have the possibility of 3 different outcomes. So the first being that, so this is your untreated, okay, no molecule or no compound. And then this is your treated, which, as you can see, the survival curve has shifted to the left. And therefore, it's an indicator that your compound is toxic. So you would throw away those compounds. Okay. The other alternative is that there's no difference between the treated and untreated. Again, useless, throw them away, or they may have other uses. Okay. So what you're aiming for is basically this sort of profile, where you see a shift to the right again in worms which are infected but treated with the molecule of interest. So this is what we did. And to cut a long story short, okay, IFAM provided us with a lot of different plant and marine extracts, which we took through the screening. And eventually, our hit was, our most substantial hit was misai kucing, or orthosiphon staminus, okay, where we showed that the active component eupatorin was able to rescue the worms from a Staphylococcus aureus infection. And how it did that was it functioned as an immunomodulatory molecule by enhancing the immune response, which was under the control of the MAP kinase signal transduction pathway. Okay, so this was something very exciting. And for Burkholderia pseudomallei, the substance was curcuma or curcumin. All right, so I think a lot of you know that kunit is a very good antibacterial agent. But Burkholderia pseudomallei being what it is, curcumin has absolutely no effect on BP. But on the other hand, it was able to enhance the worm's immune response, and then the worm was able to fight off the infection, and we got sort of similar curves as we see here for misai kucing. Okay, so this is a new technology for the search of anti-infectives, which specifically target the immune response of the host. Okay, so I'm coming towards the end. Right. So all this time, what I've described to you is a lot of work that was done on understanding the host response to infection. And then Sylvia's work was to see how the bacteria gene expression changed during an active infection. So the question now was, those were done under different conditions, under different experimental factors. So can we sort of interrogate both of them simultaneously? So what we did was we took the dual RNA sequencing approach, which is basically you have your infected mice again, okay? Now you extract RNA from the liver or the spleen or the lungs, whichever is of interest to you. And your total RNA actually contains both the host RNA as well as the bacterial RNA. So that collectively is subjected to RNA sequencing. And then the total reads that you get back from the RNA sequencing, they are mapped onto the pathogen genome, as well as mapped onto the host genome separately. Okay, then what you get from the host genome, you can do comparative transcriptomics against your control, which is the uninfected. And similarly with the pathogen transcriptome that was obtained during an active infection, you can compare it back to the transcriptome of the bacteria grown under lab conditions and so on. And then we can get an idea of at any one point, okay, what is the bacteria doing? What is the host doing, right? So this project was recently completed. But as we did earlier with the mouse analysis, we also took this further. By looking at diabetic mice. So this is an ongoing project right now. So we are interested to understand, again, at any one point under diabetic conditions, what is happening to the host and what is happening to the bacteria. Okay, so the final study that I want to tell you about is very quickly, it's on Burkholderia pseudomallei biofilm. So around about 2013, 2014, we were invited by Prof. Putucherry from University Malaya. So she had a collection of Burkholderia pseudomallei isolates, which they discovered produce different amounts of biofilm. Now, biofilm is an extracellular matrix that a collection of bacteria will produce to sort of shield them from stressful conditions such as antibiotic treatment, the immune response, and so on. So she asked, you know, okay, take all these isolates, do whatever you want with them. So what our interest was to find out what genes were involved in the formation of Burkholderia pseudomallei biofilm. So we picked a high biofilm producer and a low biofilm producer. So you can see one that does not produce biofilm and the one that produces a lot of biofilm. Extracted RNA, did comparative transcriptomics, okay, and analyzed the data. And we also were interested to see if there was any difference in virulence between a biofilm producer and a non-biofilm producer. And lo and behold, yes, there was a significant difference. The BP that produce a lot of biofilm are able to kill mice very much faster than the same strain that does not produce biofilm, or the same species that does not produce biofilm. And this was the first time Burkholderia pseudomallei biofilm was identified as a virulence factor. Okay, so based on the transcriptome analysis, we were able to propose this schematic on how these genes are involved in the production and development of biofilm formation, as well as how they contribute to the virulence that we observed with the higher biofilm producer. So carrying on from this, under a totally different project, where this project was actually interested to understand the changes in the bacterial expression profile, because we know that this bacteria originates from the soil, and then it goes on to infect humans. So we were trying to figure out what changes happen in the bacteria in terms of its expression when it transitions from the soil into the infected host. So because we didn't actually have any project funds at that time, so this is a very small project where the host in replacement of the host or animal host, we use human plasma. And the transcriptome analysis showed that when, again, out of the 181 genes that were upregulated when the bacteria was grown in plasma, almost a quarter of them were associated with biofilm formation. And this was validated when we did a biofilm assay. So when you grow it in plasma, you get nice biofilm. When you grow it in soil extract media, you don't get any biofilm at all. Okay, so the fact that biofilm is popping up all the time in our different analysis. So we now have an ongoing project where we are trying to understand or identify what are the actual protein components of the BP biofilm, and to correlate to see if we get the same proteins. Okay, similarly, we know that within biofilm, you also have something called extracellular DNA. So we want to determine if the extracellular DNA that is present in the biofilm is part of the genome of BP, or is it totally different DNA which is produced? Okay, and then eventually, the proteins that we identify will be taken through an in silico screen with our collaborator, Xavier Chi, who will be screening a library of small molecules to see any that will bind to the proteins that we identify, okay, in the hope of identifying some inhibitors of biofilm formation. If we are able to inhibit biofilm formation, then the treatment with the antibiotics will be significantly improved. Okay, this bacteria is now developing a lot of antibiotic resistance, making it even more difficult to treat melioidosis patients. Okay, so sorry if I took too long, but that's about, I haven't, it's not an exhaustive study of everything that we have done in UKM, but it more or less sums up a lot of the work that was conducted over the last 29 years. Okay, and for that, I have to thank my collaborators. There's lots more collaborators, but I have zoomed in specifically for those from FSC, okay, who have helped on all the various projects, long-term, new ones, okay, and funding from the different agencies, okay, and a lot of these projects would have been basically cut short, okay, because of different problems, different issues, if it were not for the tremendous help and assistance provided by Tuan Haji Jasmali, okay? And as usual, none of these studies can be done by one person. So I am indebted to all of these people who have passed through my lab, okay, over the last 2.5 decades or so. And I thank you very much for listening.
Thank you, Prof. Sheila, for the stellar presentation and your work, stellar work, which really explains the outstanding number of awards that you have received. And your presentation actually took us through the whole evolution of your 29 years in UKM, reinventing your research and yourself as a researcher. And now, let's get into let the Sembang Santai session begin. So we have about 79 people here, actually 77, because 2 are hosts. So 77 of you here. And I'd like to open this session. It's a free and easy session, yeah? So let's, if you have any questions for Prof. Sheila, please go ahead and ask her. And, let's learn more from one another here.
I'm happy to answer anything under the sun, even if it's not related to what I've just talked about.
Hi, Prof. I have a question here.
Yeah, please do.
Okay.
Please go ahead. Yeah.
Hi, Dr. Salma.
Hi, Prof. Hi, Prof. Sheila. Yeah, actually, it's no questions, just to say, congratulations for your I would say it's very huge achievement. Okay.
Thank you. Thank you.
Yeah, and what Prof. Kalai said just now, I can see your very smooth evolution on your research, the term evolution. So I think it's very adored with your research, and you say it's very interesting and very very nice and very in terms of the how smooth is your research. And I think that's all. And congratulations, Prof. Yeah, I adore with your research actually. Yeah, that's all. Yeah.
Thank you so much, Dr. Salma.
Yeah. Anyone else? Any students, collaborators, past, present, and future? I know those who want to still work with Prof. Sheila. She's still available to work with other new researchers, I'm sure. Students here who want to pick her brain for your work.
Hi, Prof. Farid ni.
Yeah.
Hello, Farid.
I think I'm very thankful for the motivational talk, and I would really grateful if you can share if you have any tips when you supervise your student for all the work.
That's a bit of a broad question. I'm sure if you ask the students from what 10 years ago, you can ask Dr. Shikin. She's online here. Okay, inevitably supervision ends up with students crying. But that hasn't happened recently. As one gets older, one gets milder and more tolerant. But no, my first sort of point is that I don't go searching for students. I prefer students who come to me and ask that they want to work with me. So it means that they have shown an interest. They know what they want to work on. And basically, when you know that the student has interest in your similar interest to you in your subject matter, then the supervision as well as the student's interaction with you is going to get much smoother, much easier. Does that help, Dr. Farid? Nak lagi ke?
Kalau ada.
Huh?
Kalau ada lagi, tapi
apa lagi
terima kasih banyak, Prof.
Yalah, tapi I think I did have some sort of advantage as well, which was either directly or indirectly, I was able to provide most of my PhD students with attachments in foreign labs. So when they went over to foreign labs, these were in the UK, in the US, in Japan, Sylvia went to Portugal. So they had the ability of experiencing what research is like in a non-Malaysian lab, okay? And then they can appreciate, they probably appreciate the fact that here in Malaysia, we may have more things accessible to us, okay? Not necessarily equipment or chemicals arriving the next day and so on and so forth. But maybe the lab atmosphere is much nicer in UKM as opposed to in Stanford University or in, where else. So I know now financially, there are not all that many opportunities for students to go on research attachments during their PhD program. But I would advise students as far as possible to try and take advantage, look for them specifically, don't wait to be told about it. Look for it specifically, okay? Try and get the experience of working overseas.
Okay, Prof. Sheila, we got a question from Shuhaila. She comments, Prof, very inspiring, but may I know how Prof finds the path on expanding your research project? How do you find your path or your direction in expanding your research project?
Is that from now or from earlier?
I believe, Sheila, you're an early researcher, right? Yeah. So I'm thinking from the very beginning. How did you start off?
Hold on, let me let this guy go off first. Okay, so thank you for that question. So I hope you could see from what I said during the earlier part of the talk is first thing is, you join an existing group, a well-established group, so you don't have to basically start from scratch in terms of getting access to facilities, getting access to research funds. Then that will help to establish yourself in terms of publications, in terms of your skills. And with that on your CV, you are then able to go on and get grants yourself. But then I guess the question is, how do you move from where you started to where you are right now? So a lot of it was evolving with the times. Okay, so as I mentioned earlier, the first phase was working with hybridomas to produce monoclonal antibodies. We needed monoclonal antibodies, okay? But if we were to continue down the lane with working just with hybridomas, anyone who's worked with cell culture will know the immense problems that you can find. First of all, contamination. Second of all, if your freezer breaks down, then that's the end of your 3 or 4 years work. You lose the viability of the cells, even though they're immortalized and so on. So every time you progress to using the latest technology, especially when it's a molecular-based technology, you know that you have certain advantages that will cover you for quite a while. So when we switch to making recombinant antibodies, as long as you have the phage clone, right, you can just retransform it into E. coli and tell the E. coli, hey, produce these antibodies for me, right? It's so much easier. So same way, I guess I was lucky in terms of being invited quite a few times to propose projects, okay, to build up, that ended up building up my expertise and my path, research path. So if you have the opportunity to do that, please, follow that. Don't get stuck in sort of one dimension. Think out of the box and keep moving forward.
Okay, thank you, Prof. Sheila. Another question from Yip Chi Hu. What would be your take-home message to young researchers to do good science like you did?
After spending 6 years, 7 years in my lab, you're still asking me that question, huh? Anyway, so hang on, what's the most challenging thing that you have to face? Most challenging thing, I presume, is, of course, now it's a case of finding funds, okay? The whole way of doing research has also changed over time. So in some ways, I'm lucky that I'm at the sunset end of my research career, so to say. Things were much easier before. But, when you don't know how easy it was before, so there's nothing for you to look back on. You just keep moving forward. And any challenge that you face, there is always a solution. If you can't do it yourself, get help. I think that's what I have to say.
Okay, thank you, Prof. Sheila. There's a couple of more questions from Azrin. Azrin says she admires your work a lot from her years as a degree student here in UCAM in 2016, and just would like to know what are the challenges that you have faced in your career and how you overcame these challenges over the 29 years?
So I think a lot of it I answered through Chiho's question, but thank you, Azrin, for the question. The other thing is that as a scientist, you know that you have to evolve, you have to move with the times, and you basically have to be open to all possible opportunities that are out there. I mean, if you don't try, you won't know. If you do try and you fail, it doesn't mean that you stop. You keep moving forward. Okay. Prof. Firdaus and I have, submitted so many grants to MRC, to Wellcome Trust, and each time we've got rejected. But that hasn't stopped us from, trying alternatives or improving on the previous proposal, and trying to see if we can succeed, even if it's a small grant. Small funding doesn't mean the outcome is small also. It all depends on who is working on the project, how what's the word, how independent they are, and how they can overcome these issues that we face in that particular project. Okay, so it's collaborative. Don't do anything on your own. Don't be afraid of asking for help. Don't be afraid of seeking advice from those who have been through that same issue before. That's the only way to move forward, to keep moving forward.
Okay, thank you, Prof. Sheila. Another question from Dr. Farid. He wants you to glance into your crystal ball and tell him, what do you foresee for the next 5 to 10 years with regards to research progress in BP?
Thank you, Farid. I think the main focus right now is more on vaccines. Because, as I said earlier, Burkholderia pseudomonas is developing resistance to a lot of the antibiotics that currently work to kill or eliminate the pathogen. So it's becoming more and more difficult to treat melioidosis patients, particularly those who have diabetes. And one of the limitations of working with BP is that it's a multifactorial bacteria, meaning that just because you design a vaccine against one particular protein that the bacteria produces is not going to shut it down, or is not going to neutralize it. So many different vaccine candidates have been developed, have been tried, tested, and none have been able to provide more than probably 50% protection. I mean, we had one particular protein that went up to 70% protection in mice. But there is no miracle one type of vaccine. So a lot of the researchers in Europe and in the US, they are currently looking at the possibility of different proteins, combination vaccine candidates, drug repurposing to find new alternatives to treat melioidosis patients. So I foresee that that's where the next 5 to 10 years is going, because in terms of pathogenesis, yes, there's still a lot of virulence factor genes which have not been identified, and that's one of the factors that has inhibited the development of a good or successful vaccine. If you identify a gene with unknown function, do you pursue it? Do you go ahead, sort of move in the dark? No, so there has to be all these parallel studies that are going hand in hand. Some groups trying to identify what these proteins do, and then other groups trying to develop the vaccine strategies for melioidosis.
Thanks, Prof. Sheila. We've got a question here from one of your co-researchers, Dr. Shazila. She wants to know your routine in writing papers and how do you cope with your writing amidst all the other duties that you have as a lecturer, researcher, and supervisor.
Shazila, mana lah you dengar ni? Tulis dari 7 pagi sampai 9 pagi.
Betul lah nak tahu Prof. Betul ke rumours tu?
Tak betul lah. No, how do I do it? A lot of it has to do with the students. Okay, the students have been very resilient, as I said, because they came looking for me, so they have high interest in the project that they undertook, and so they were very happy to draft the manuscripts, which made my job very much easier, to basically polish it up and get it to the standard of the journal that we had intended on submitting it to. Yes, I did write a few review papers myself, but basically, you just make time, because when writing review papers, you have to do a lot of reading, and you have to be systematic, because you can't rely on something that you read 6 months ago, which that was interesting, but where is that paper now? What was the actual fact that was interesting enough to put into this review paper? Yeah, so it's just being disciplined about it. Okay. I know I have friends overseas who are scientists who work only from 9 to 5, but they are very prolific writers. So there's nothing to say that, oh, you have to work overtime all the time in order to come up with the publications that you need.
Thank you, Prof.
Okay, Shazila, got it?
Okay,
Right.
okay.
All right. I think we have the Spice Girls in our midst. There's a person called Mel K. I've always heard of Mel B and all the Mels, but not Mel K. But today we have a Mel K who wants to know, what would you have done differently in your career? Is there anything you would have done differently in this if you look back at your 29 years?
I'm not sure. It's not that easy a question to answer. I've always, it's not always, but I have asked myself every now and then, what would have happened if I had stuck to DNA repair and not moved to become a mock microbiologist? So yeah, I don't really see anything different that I would have done. Okay, maybe to be frank, I was more introvert than I am now. I'm still an introvert, but I was extremely introverted during the first, say, 10 years of my career. So I could have taken more opportunities that were available, but sometimes you just don't want to do it because you have to talk to a lot of people, okay? And that's not easy for some of us, or you're not ready to face rejection in terms of grant proposals and so on. But when you start interacting with others, in-house, other people in-house, and you learn from them, and then gives you more strength, builds your character to sort of move forward, take up the opportunities that are available, sort of thing.
Okay, thank you. It sure doesn't seem like you have any regrets, because you've moved along very nicely, very smoothly through your 29 years here in UCAM. One more question. From all the projects that you've been involved in over this almost 3 decades of research, what would be your most memorable research project? Something that, if you say pick one, which project would it be?
I guess it has to be the Stanford project. No offense, Sylvia, and the rest of them who are here. But with the Stanford project, I had a group of students who were really, enthusiastic, really enjoyable to work with, and because I guess we had the perks of extra funds, a lot of funds in fact, which enabled us to do a lot of things and get a lot of results.
Okay. Just one more question. I mean, to the young researchers and those who are just beginning their career here in UKM, what would you state as your key characteristics or key values that you need to have to be a resilient, successful researcher in UKM?
Simple, you have to be passionate about your science, that's it.
Okay.
It's not a job, it's part of your life.
Right. Thank you. But we have one more question, and someone's already wanting to, wants to know who's going to inherit the tiny worms.
Dr. Farid, you nak buat apa dengan cacing tu? Well, I don't know.
What going to do with the worms?
It'll be there. It's a question of, unfortunately, there's nobody who is trained to work with the worms, who is around. But, you know, we've worked with Umbi got the worms from us. So there is a possibility that the whole stock could be transferred to the C. elegans lab in Umbi.
Okay, any more questions from the floor? Okay, you can always type in, yeah, okay, yeah.
Okay. Hi, good morning, Prof.
Good morning.
Shikin here.
Hi.
Okay, I'd like to take this opportunity to personally thank you. Okay, I'd like to personally thank you for your guidance, support, care throughout my studies in UKM as well as in UM. So it was a pleasure to have you as a supervisor and co-supervisors that shaped me who I am today. So I wish God grants you health, wealth, and happiness throughout your life. Thank you.
Thank you, Shikin. The pleasure is mutual. Hello, Prof.
Hello. If there is no other question, I'm sure there are ton of questions that have people around has their question in mind. But before we stop, or if any question, you are welcome to the floor. However, if the end of the discussion, again, like mentioned by Prof. Kalai again, and also Prof. Sheila, this talk actually meant for everyone in our jabatan or else is okay, because we just sharing what we have, what experience we went through for last how many years, including me and others. Probably we can have some sort of guide. It's a little bit of guide, not 100% guide, especially the young one. That's why the program, the talk has been divided by 2 parts. Now the one first 6 months is kind of senior, and the next 6 months will be kind of junior. There's nothing at the middle yet. So my advice is for the younger one, for the next 6 months, please don't feel bad if you thought that you are at the early stage of your career, because just don't make comparison for the next 6 speakers. So my advice is, please, those that for the next 6 months, this is 3rd month, 4th, 5th, and the 6th, then there will be another 6 months. 6 months will be the younger one. So for the younger one, again, don't feel so bad if you think that you are kind of early. That's okay. There's the the path that we have to take. Partly, probably Prof. Sheila already mentioned earlier. Sometimes it's just a luck that we went through it. Sometimes we make a good choice, but however, don't feel bad, because you, Prof. Sheila and me, we went through it for the last 20 and 30 years. So I think my advice is for the younger one, for the next I mean, after June or July, those that probably have prepared, names already given to the, all of you, I mean, all the 12 of you for this year. So don't, my advice, please don't feel bad if you are already started. For the, I don't call the old one, the senior one, please, share whatever we have. Probably our younger one could learn something from us that may kind of guide them to somewhere. At least, I don't know to where, but at least somewhere. So that's it from me. Probably if Prof. Kalai want to open it to the floor again, go ahead. But if this is the end, probably, but not the end of that, because we're going to see Prof. Sheila around, all those us around, then ask question again and so forth. So that's it from me before we end the the Sembang Santai. Thank you.
Wani, you're muted.
Yeah. Someone muted me, quite. Hi, Prof. Sheila. What are the differences that you can see in terms of students, research, grant, KPIs that we see here in our as from the start of your career to now? What are the differences that you see? I think it's a very simple question to answer.
Hi, everyone. Differences. Well, of course, things are a bit more stringent now in terms of KPIs, as you mentioned. And also your independence is a bit limited in terms of doing your research.
Can Suleimani mute yourself?
Students, I mean, it's hard to tell. You had students who didn't complete their PhD or their masters, 15 years ago, for one reason or another. So it doesn't mean that the students before were different from the students today. You have a lot of enthusiastic students now who are very capable, highly capable. A lot of them have been through my group. I know that whatever they put their minds to, they can achieve. So I don't think there should be any problem for your next, 30 years?
Okay, one last question from Shohaila again. Yeah, she wants to know if you've ever faced difficulty where your research work never succeeded or didn't come up with the results that you planned for. How do you stay motivated and positive when things just don't work out?
Bit of a difficult question. But the thing is, it's very rare that you only have one particular project running at any one time. Okay, so even if it's a whole research project, you could have sub-projects, some which are producing beautiful results, some which are having difficulties. That's only to be expected. So there's always something to counterbalance the down that side that you feel at any point, because you've done multiple experiments, all of them have not been able to produce the results that you want. But you just persevere. And as I said earlier, you can't just continue to think along the same lines. You have to expand and see, maybe you don't know the answer to troubleshoot. So ask somebody else. Chiho can attest to that. We had a lot of problems because his was a very difficult project, but we got help from from our collaborators in the UK. So eventually, everything worked out fine.
Okay. I don't see any more questions in the chat box. Unless anyone has any burning questions to ask Prof. Sheila at this point, can you unmute yourself and ask her straight away? Or we will come to an end of this session now. It's already 11:34. Any other questions? Going once? Going twice? Going thrice? Okay. If none, I'd just like to take this opportunity to thank Prof. Sheila for availing herself to give us this, this run through of her 29 years here in UKM, with showcasing all the excellent work she has done, all the excellent connections she has made, the students that she has trained through her labs, and I'm sure some of them are here, and also the excellent networking, local and international, even the people that she has worked with here in UKM. Extraordinary work and very elegantly presented. So we just want to put our hands together and thank you, Prof. Sheila, for your presentation this morning. Let's all applaud, yeah, her presentation for this morning. Right, thank you.
Thank you,
Thank you. All right, bye.
Thank you, everyone,
Bye-bye.
for coming.
Thank you. Bye-bye. Have a safe week and weekend ahead. Yeah. I'm going to end the session.
dalam boleh tulis lah kalau tanya bukti dah bukti. Ada dah sehari 11.38.
Ini tau, bicara. Macam, alah, dia tak daftar lah saya, dia dah tengok.
Sikit. Macam mana ni? Dia dah ada.